So in this video, we are going to look at DNA replication, which is basically the making of DNA. And remember if you looked at the other videos that I have on DNA, I do everything in threes. DNA has 3 letters. It stands for deoxyribonucleic acids. And we know that DNA is our cookbook. And just like cookbooks hold recipes that tell us how to make things, that's what our DNA is. Our DNA is a huge cookbook that holds genes. And genes are recipes that tell our bodies how to make proteins. And when we look at DNA, again, the rule of 3, DNA has 3 letters. DNA is put together with building blocks called nucleotides. And those nucleotides have 3 parts to them: a sugar, a phosphate, and a nitrogenous base. And in the case of a cookbook, you have covers and you have pages. And you know that the covers are there to protect the pages on the inside, because the pages are the important part. It's the written word. It's the part that you want to read. Well, that's how DNA works as well. The sugars and phosphates make up the sides of DNA, and they are your book covers. The sugars and phosphates are not your genetic material. Your genetic material are your nitrogenous bases, your A's, your G's, your C's, and your T's. And if you remember the base pairing rule, which is also called Chargaff's rule, A's and T's always go together, and C's and G's always go together. So when you open up a cookbook, you have a recipe on the left side and you have a recipe on the right side. Well, your DNA is the same way. When you open it up, there's a group of letters on the left side and there's a group of letters on the right side. And these are your genes. These are the recipes that our body read in order to make our proteins. Well, whenever you need to make a new cell, you need to do what's called DNA replication. And the only time a cell should make new copies of its DNA is during cell division. And cell division is mitosis and meiosis, both of which I have videos here on YouTube about. In the case of mitosis, this is where our bodies make clones. Like you've cut yourself, or your cells just die and you have to make new cells. So if your skin cells die, you need to make sure that you have new skin cells being made. If stomach cells die, you need to make new stomach cells. So in mitosis, you are making what we call body cells. So you need to copy the cookbook so that every cell has the recipes it needs to do its job. Meiosis is the making of egg and sperm. And it also will do DNA replication, but there's a few twists and turns to that. So like I said, I do have a video on YouTube about mitosis, and I have a video on YouTube about meiosis. So if you need information on that, you can check those out. But whenever your body needs to make a new cell, it needs to make an exact copy. You can think of it this way. You're about to have a child, and you want that child to have every single recipe it needs in order to be successful in life. Well, that's what your body's doing. When we get ready to make a new cell because a cell has died, a cell in that area will go through cell division and it will copy its DNA. So in the end, we end up with 2 cells that are genetically the same. Now, again, like we mentioned, remember DNA does have 2 sides, and so we have sugars and phosphates, sugars and phosphates. So the blue and teal here would be like the book covers, and the different color puzzle pieces in the middle are our pages. And again, with Chargaff's rule, or what we call complementary base pairing, T's and A's fit together like pieces of a puzzle, and C's and G's fit together like pieces of a puzzle. So if you were to break those hydrogen bonds, which are the dotted lines going down the middle, remember, we keep books closed in order to protect the pages so the pages don't get damaged. Well, our DNA needs to stay closed in order to protect those pages, and those pages are our genes. And so hydrogen bonds are kind of like the zipper that keeps our DNA closed, because you wouldn't want your book to fly open by itself, because then the page can get torn, the page can get stepped on, the page can get smeared. Well, those are mutations, and we're going to talk about mutations later in this video. Well, remember also with DNA replication, if you know one strand, you know the other. So here I have a piece of DNA. And if you remember again the rules of 3, every 3 letters on a piece of DNA is called a triplet. And so just like you learn to read a sentence, genes are sentences. And just like you know a sentence starts because it has a capital letter, and you know a sentence ends because it has a period, our genes are the same way. our genes will start with a capital letter and our genes will end with a period. Because somehow our bodies have to know, okay, where does the recipe start and where does the recipe stop? Well, we have learned how to read the sentence of our DNA. Just like when you were little, when you first started looking at books, you would recognize straight lines or you'd recognize, curves. And then all of a sudden one day, you knew that this was an A, or you knew this was a T. And then all of a sudden, those T's and A's and C's came together to make a word, cat. Well, our bodies do the same thing with a gene, which is what we see here. But in order to read it, we have to know, okay, let's break it up into words. So our bodies take a gene and divides it into groups of threes. So ACG would be one group, TAG would be another group, but then GC, there's no third letter, so your body ignores it. Well, every 3 letters is called a triplet, and it tells us one ingredient. Remember, a gene is a recipe, and it's basically a chain of ingredients that we put together in order to make proteins. And remember, the ingredients we use to make proteins are called amino acids. So if this were a recipe, I know that this recipe only needs 2 ingredients because I only have 2 triplets there. Well, if you remember complementary base pairing, if I know one page, I also know the other page. Because on the other side, I would have T, because T always goes with A, then G, because G goes with C, and then so on and so forth. So if I were to ask you to write down what the other side looks like, you would put T, G, C, A, T, C, C, G. Now, when DNA replicates, we're going to photocopy both sides. I know a lot of people have printers at home or you just take pictures of things, so Xerox machines might not be something that you're quite familiar with because a lot of people just don't have them anymore. But with a Xerox machine, I can put a page and it scans it and it copies it. Well, with DNA, think of DNA as a cookbook. Well, you can't photocopy every single page at the same time. Or think of DNA as a computer. You go onto the internet and you find a cookbook. And you're like, okay, let's print out all these recipes. Well, you can't print every single recipe at a time. Well, DNA is the same way. We cannot photocopy every single piece or every part of DNA at the same time. So just like you open a book to pages 1 and 2, you can photocopy pages 1 and 2. Then you can open it up to pages 3 and 4, and you can copy pages 3 and 4, and so on and so forth, until eventually the entire piece of DNA gets photocopied. So when DNA replicates, each side gets photocopied at the same time. And this is what we call a template. The original piece is a template. That's going to be what we use to copy the new recipes. And remember, T's will pair with A's and C's will pair with G's. So I always talk about numbers 3. So if you're holding a book in your hand and you need to photocopy that book, well, obviously the first thing you have to do is open the book. Well, that's what happens in step 1. We've got to open our DNA. But just like you can't open every page in the book at the same time, we can't open every part of DNA at the same time. So we have a little enzyme, and remember, don't panic when you have enzymes. Excuse me. The name of the enzymes, if you read them backwards, always give you a clue. Remember, anytime you have ASE at the end of the word, that's an enzyme. So you're used to reading sentences from left to right. When you look at an enzyme name, read it right to left. And what I mean by that, we have an enzyme called DNA helicase. Well, ACE lets you know it's an enzyme. Helix means to twist. And then we have DNA. So this enzyme has something to do with the twist in DNA. Well, just like if you wanted to copy pages out of a book, you have to open the book. Well, the first thing we have to do is we have to open our DNA. Now, you cannot open every page in a book at the same time. So we do not open every part of DNA at the same time. Think of it as a zipper. You're going to unzip it a piece at a time. And that's kind of what this DNA helicase does. Okay? DNA helicase will start, just think of it as starting at the top of DNA, and it opens your book to pages 1 and 2. So just kind of like the first twist. And then it will sit there and it will wait until we have a chance to photocopy pages 1 and 2. Then it will unzip a little bit more and open up pages 3 and 4. It will not go down and unzip the entire piece of DNA at one time. And again, the reason for that is you can only copy 2 pages at a time. So if I unzip my entire piece of DNA, it's like tearing all the pages out of my book. And while I'm focusing on just 2 pages, pages 7 and 8 could get lost. Pages 12 and 11 could get stepped on. And well, that's what mutations are in DNA, which again, we're going to look at later in this video. So what DNA helicase will do is it will start and just unzip a small portion. And when I talk about unzipping, it breaks the hydrogen bonds. So that allows you to open your book. And so we're going to open our books to pages 1 and 2. And then our little friend DNA helicase just kind of sits there. And so the enzyme DNA helicase will unwind the DNA and break the hydrogen bonds between the DNA strands. This is called unzipping the DNA. But again, it's only going to do just a small piece at a time. It would just be like you opening it up to pages 1 and 2. Now, we talked about how the building blocks of DNA are these things called nucleotides. And I always draw nucleotides as letter L's. Well, just like you're cooking and you start a recipe, you usually make sure you have all of your ingredients before you start the recipe, because you don't want to get halfway through the recipe and realize you're missing an ingredient and have to go to the store. Because if that happens, your product, whatever it is you're making, could be totally messed up. So your cells actually have extra nucleotides floating around because you don't want to start the process of DNA replication and say, oh, I've got a T here. Can somebody go make me an A? You don't have time to sit there and wait for that letter A to be made, because during that time, you could have a mutation. So as soon as helicase unzips a portion of our DNA, a second enzyme, DNA polymerase, comes into play. Again, look at the name. ACE means it's an enzyme, right here. ACE means it's an enzyme. A polymer is a molecule we're putting together. Poly meaning many, so it has many parts. So this is an enzyme that helps put together DNA. So once we open up our DNA, this DNA polymerase makes sure that everything gets mixed together. So we always have free nucleotides. These are the extra ingredients we always have in the nucleus. Because again, it's better to have it and not need it than need it and not have it. So as soon as we open up the book, we immediately start to copy the recipe. And that's what's called base pairing. So DNA helicase is open the book up to pages 1 and 2. And then we're going to photocopy pages 1 and 2. And that's the job of DNA polymerase. DNA polymerase will scan the pages and say, okay, there's a T, let's stick an A. There's a G, let's stick a C. And it will go through and it will line, as you can see here, it will line things up. So wherever there's a C, there's going to be a G. Wherever there's a T, there's going to be an A. And DNA polymerase's job is basically just to match things up. So it's like we're photocopying these pages. Well, when DNA polymerase kind of runs into DNA helicase, that's DNA helicase's cue to go and unzip the next section. And then DNA polymerase will go down it, pair everything up. It's going to hit DNA helicase, and DNA helicase unzips the next section. So eventually, we will unzip all of DNA, and we will photocopy every single page. But then something's got to put everything back together. And that's going to be the job of this molecule called DNA ligase. Step 3 is called bonding. I call it putting it all back together. So again, if you have ACE, that lets you know that you have an enzyme. The root word lig or lig means to glue or to tie. That's where ligaments get their name from. A ligament is a piece of tissue that holds our bones together. Okay? If you become a lawyer, you'll do litigation. That's where you take all the pieces of the story and you tie them together. But with DNA ligase, its job is basically to glue everything together. The formal definition is to link the nucleotides using covalent bonds. So as you can see in this picture down here, right here would be where DNA helicase is. It has unzipped to that point, and then going down, we have the DNA polymerase. It's going to be making sure all the pieces come together. And then I have to have my stapler that's going to put, or my glue, that's going to glue everything back together. So DNA helicase comes first. It will just unzip a piece at a time, and it will wait. DNA polymerase comes, lines up my A's, my G's, my T's, and my C's. And when it hits DNA helicase, that's DNA helicase's cue to unzip the next part. And then going along behind all of them is DNA ligase. DNA ligase's job is basically to glue everything together. So in the end, I have 2 stacks of paper. And I have book covers on them, and I have glued them together, and I have 2 identical cookbooks. Because we have photocopied every single page, we make sure that everything is paired up. So DNA polymerase puts A's and G's together, A's and T's together, C's and G's together with hydrogen bonds. And the DNA ligase will come along and basically glue the sugars and phosphates together to make the hard book covers. So in the end, we have our 2 books. So our results are 2 pieces of DNA, and they are exact copies of each other. And for many years, the argument was, okay, we agreed that we made new DNA, but what happened after that? Well, we now know that DNA is what we call semi-conservative. Okay? What this means is when we make DNA, you end up with 1/2 old and 1/2 new. So when we make the DNA, here's my original piece on the left, and then I've opened it up, and then I go in and I photocopy it. Well, some people thought then that the old pieces, that it came apart again, and the 2 old pieces came back together, and the 2 new pieces came back together. Well, we now know that that's not true. There are some famous experiments. One was done by Hershey and Chase, in which they took radioactive nitrogenous bases and added it to a cell, and they waited and allowed that cell to replicate. And then they studied the DNA, and basically it would be like half the DNA glowed, half the DNA was radioactive. So this is how they knew that DNA was what we called semi-conservative. Semi meaning half, and conservative means to save. So when we make new DNA, it's actually 1/2 old and 1/2 new. But like I mentioned, when we make DNA, we have to be real careful because we don't want mutations to occur. Okay? By definition, a mutation is a change in the triplets of DNA. All of your life, you've heard of mutations. Well, mutations are what run evolution, as you'll see later when we discuss evolution. A mutation is where we change our recipe. So remember, genes are recipes that tell our cells how to make proteins. Different cells need different proteins. So if you think of DNA as a cookbook, it has chapters. So just like your cookbook has a chapter on bread, poultry, soups, desserts, your DNA is also divided into chapters. And that's what makes all of your cells different. So your brain cells might use the recipes in chapter 5, while your stomach cells use the recipes in chapter 10. Well, they're different chapters, they're going to have different recipes, so they're going to make different proteins. And remember, structure determines function, so if cells make different proteins, they're going to have different jobs. So cells are very specific as to the proteins they need. Well, when you have a mutation, that's a change in the recipe, and that can cause the cell to make the wrong protein. And if the cell makes the wrong protein, not only is it wasting time and ingredients, if it's not making the protein it needs, that cell could die. So a mutation is where we change a recipe. So like I said, if you go to the store and you buy a cookbook, you're going to trust that those recipes are correct, and you're going to follow that recipe. So you're making a chocolate chip cookie, and you're following that recipe. But when you bite into it, it's nasty. Okay? Well, you trusted that recipe. You put a cup of salt in it, just like that recipe said. You had no way of knowing that it was a mutation. You had no way of knowing that it was supposed to be a cup of sugar. Well, your body's the same way. When a mutation occurs, your body has no way of knowing that this recipe has been messed up. Now, in our bodies, there are two types of mutations you can have. You can have what's called a gene mutation, or you can have a chromosome mutation. And later on, we'll talk about the difference between genes and chromosomes. But a gene is just one recipe. So if you have something called a gene mutation, just one recipe in that entire cookbook is messed up. Something like diabetes is a gene mutation. Hemophilia is a gene mutation. When we have chromosome mutations, entire chapters of a cookbook are messed up. Something like Down syndrome or Huntington's disease. So what we're going to look at today are what are called gene mutations. Now, one type of gene mutation is called a point mutation. And a point mutation, basically just one or two letters are changed. So it would be like me changing the recipe from saying sugar to saying salt. So we still have the same number of triplets. As you can see here, point mutations are also called substitutions. So my gene is supposed to say the fat cat ate the rat. So remember, those are triplets. So 1, 2, 3, 4, 5, 6 triplets. So it's basically telling me I need 6 ingredients. But notice one letter changed. That's what's called a point mutation. So now the sentence reads, the fat hat ate the rat. Well, that sentence doesn't make sense. Well, that's what a mutation is. You're going to make the wrong protein. Your body will still make this protein. It'll still make a protein with 6 ingredients, but it's not going to have one of the ingredients is going to be wrong. So therefore, it's going to make a protein it cannot use. And if it's an essential protein, that's enough that that cell could die. So this is a gene mutation. Just 1 or 2 letters get substituted. They get messed up. Now, another type of mutation is called a frameshift mutation. With a frameshift mutation, this is often called an addition or a deletion. What happens is a nitrogen base gets removed or added. So in this picture, this is an example of what's called a deletion. Notice what the gene is supposed to be. ATG, GAA, GCA, CGT. Well, notice the G got deleted, but we still need groups of 3s, so everything shifted. So notice my recipe is supposed to have 4 ingredients. Now my recipe only has 3 ingredients, and they are the wrong ingredients. So this is an example of what's called a deletion frameshift mutation. It frame shifts because all the letters shift because I still need groups of 3. The same thing could happen with what's called an addition, where a letter gets shoved in there. It's still going to push everything and move everything out of the way. So when you have a mutation, you are making the wrong protein. You change the ingredient. But you're going to see that not all mutations are bad. There are things called neutral mutations. A neutral mutation occurs when we change, it's a type of point mutation. It's when you change the letter, but the ingredient does not change. So that's DNA replication.